it's a good trait. And when we look into this family, we see a lot of different snakes, and they are super diverse and have a lot of different variations. So we have some that have a rattle to make some noises to warn others. Others are, or the venom was used to make some important drugs. Other form the kingdom of a lonely island where they are the ruler. Others are named like a metal band or like the lead singer of a metal band. The Gaboon viper has one of the longest fangs, up to 4 centimeters of all snakes. Others looks like a more or less unicorn, but with 2 horns. Some are named like persons out of the Harry Potter universe, or even have a modified tail that can be used as mimicry for spiders to catch some birds, and others even live into the far north where other reptiles are not even able to survive. So I think they are super awesome and super diverse. The only drawback is that a lot of reptiles are endangered to be threatened, and so when we have no conservation, there will be no variation that we can study. And 50% of the snakes are endangered, and of the Viperidae, so there are around 300 to 400 different species. And the study showed that around 10% the data are not enough that we can say anything about them. And here you can see that nearly 20 or more than 25% of the vipers we know something about are endangered, and that makes them to the second most threatened snake family when we look into all the others. And that is important to say. So without the conservation of animals, we don't have the variation to investigate them and make some awesome findings and investigations in these animals. But now let's talk about the venoms. So some vipers, some Elapidae, some Colubridae at some point developed the venoms. And the venom is composition of different proteins, peptides, and over the time of the evolution, some proteins were used from another organ, some other function to be included into the venom. Then the speciation started, and so we have now a lot of different species with a lot of different venoms. And one major aspect for the snakes to use this venom is to use it for the hunt. And so, for example, for the North American pit viper, they could show that the more diverse the diet is on the salinity level, the more complex is also the venom, which means if we think about venoms as some kind of cocktails the snakes need, it's always about what will they eat. And so cocktails, or the venom composition, is more or less like a cocktail, and the different ingredients are the toxins. So if you prey just on a small variation of prey, then it's not that needed to be to have a highly complex venom. But if you hunt for different kinds of prey, so for rodents and for insects or something like that, then it might be useful to have a more complex composition. And if you are more the all-rounder and eat everything that you can see, it might be even more helpful to have a lot of different toxins that might be venomous for different kinds of prey. And so when we look now into the different cocktails of the different families and subfamilies, you can see that on the proteome as well as on the transcriptome, different toxin families are more abundant in one or in the other group of snakes. The elapids are mostly composed of some kind of peptides, while pit vipers and true vipers show more focus on phospholipases, metalloproteinases, as well as serine proteases. And some similar you could also see in the transcriptome, where the small three-finger toxins were also more abundant in the elapids. And so one of my the main topic of my PhD thesis, then I want to look into more into the cocktail of the true vipers. and want to see what is really in. And so I started to focus on the outward vipers, so the subfamily of Viperinae. Over 100 different species can be found, and one are ruling large kingdoms, so they can be found from Britain up to Japan. Others are nearly everywhere on Africa, and on the other side there are so variants that some species are just endemic to small islands. And the venom is composed, when you sum all up, over 16 different toxin families, where they can be subdivided into major toxin families that can be found in nearly all of them. Secondary toxin families, also highly abundant, but not to be found in each of the species or genera, and some more minor toxins and even some rare toxin families beside peptides that can be found in all of them, but are not that much investigated. And you can also see that the variation of the different toxin families is high, but also the mass range. So some of them are up to 100 kilodalton, and these are just the monomeric sizes. So if you now start to combine them to dimers, multimeric construction, then you can even go higher. But on the other side, you have small peptides that are just made out of 3 amino acids, or shorter peptides like the Kunitz type inhibitors at around 6 to 7 kilodalton. So even for the analysis, you have a big range that has to be covered if you want to investigate all of them. And I'm mostly focusing, thanks to great cooperation, on Turkish vipers, where a lot of these old world vipers are present and where I could analyze plenty of them. So a lot of them I could analyze. I'm really happy about that. And when we look more on the old world vipers in general, so less than 50% are studied until now, more than 20 different toxin families. And when you look here, all the red ones are species where the venom was at least once time investigated, and you see most of them is still unknown. So there's a lot of venom that we can analyze, a lot of hidden treasures we want to find. And when you sum up everything, there are some smaller, or you can go into the genus, and then you see that there are some trends you can follow. But since this table is a bit more chaotic, let's go a bit more into the sum up versions. So one of the analyses you can make to investigate the snake venom is the snake venomics approach. This classic one is first made by an HPLC, then by an SDS page, some digestion, and then the investigation by mass spectrometry, and then finally the quantification. And you see that like the beetles has some more focus on nearly all of the toxin families, while the Vipera genus has no disintegrants, and so you see that the more you go up, the less diverse the sum up composition goes. And if we place now all the made compositions beside each other, so they are all analyzed by the same approach, so they are nicely comparable, you can see some trends, but it's easier if we make it a bit broader and show the different toxin families side by side. And then you can clearly see something like here, the phospholipases are here more abundant, but therefore these snakes have a lesser amount of metalloproteinase in them. So it seems like that there's a trend to have either this or the other one in the venom. And the dotted lines here are always differentiated, different genera from each other. Other remarkable things is that also the snakes that has less metalloproteinases have also less disintegrants in, since they are mostly coming from one type of metalloproteinase. It's not that much surprising, but some of these Vipera here has more metalloproteinases, but no disintegrants, which means they have a different kind of metalloproteinase in their venom. Venom growth factors can just found in this clade, with a small amount in some other beetles. And there are some outliners, like this is 13, that is just found in some beetle species. Here are some daboyas that are also a different species. So you can see here within one species, within one genus, there are different combinations, but with it I will talk a bit later about. And here also within the beetles, there's clearly one outliner, while all the others have more metalloproteinase-based venom. This one is clearly focusing more on the phospholipases. And one part of my PhD thesis at the moment focusing on even more Turkish vipers, and therefore I want to show you a bit more about their composition. So this is a subpart of the current project. It's about the Montiviperas. They are closely related, can be found nearly everywhere in Turkey, but with distinct areas for different species. And even evolutionarily, they are not that far away. So they are all doing less than 10 million years ago. And so I investigated the Montivipera burgadacica, also their subspecies Albizona and the Montivipera xanthina, which are marked here so that you can follow the tree bit. And there was one previous study about the Montiviperidae. So all of the snakes have more or less the same diet. They are close related living. They have some more or less similar habitats in that they are living. And so even the venoms are highly similar. So even if I compared with a study that was made 2008 on the Montivipera, now with my 3 new investigated compositions, they are similar. They are highly similar. The HPLC chromatograms show some differences. So there are some proteins that might be different, but the overall composition are really similar. And when you look into the diet, that makes also to some degree sense. And the other genus I want to talk is about the viper. So they're also close related. And if we look here, so we are also just around the 5 million years ago. And when you then look more into the Peleus group of the Vipera, not the Vipera per se, but the Peleus group within them, then you see they are also even more diverse in their distribution. So we have some in Europe, then up to the Caucasus, but also in Russia, Georgian, Armenian, etc. So they are not that close related from their distribution, but from their genetic distance. And there some venoms were already investigated, marked by the black stars, and I looked then more, again, into new ones that are from Turkey, the Viperidae that was found there, and the Viperidae. And when we compare now all these compositions, it shows that they are not that similar like it was in the Monty vipers. So I find most remarkable the Viperidae, which has nearly none metalloproteinases in. And the Dareski fits into the composition that was also investigated from the Katsnakovi. They're also relatively close related. And the subspecies from Anatolica and Zenliki show here the highest grade of metalloproteinases in the venom. And so you can see there's some kind of trend that if you follow the tree into that direction, the amount of metalloproteinases increases, while the serine proteases roughly decrease to some part. Yeah, that's about the venom compositions and what I am looking at the moment into. But there are a lot of more variations in the methods. So they can be roughly, well, they can divide into bottom up and top down. So there are some HPLC separations that can be made. Then you have a separation by SDS gels, 1D, 2D digestion. Mass spectrometry is a major key technology that is used. Or you try to investigate the toxins in their total, so no digestion. You try to get the analysis of the toxin directly like it was in the venom. And then you need the power of computer to analyze them, to compare them. So there are a lot of combinations that can be found. So you don't have to make the HPLC first. You can also just run one SDS page. And so you see the method that you can use just for the comparison, for the analysis, is also that variant like the venoms are that they're used to investigate. But maybe I explain it a bit easier and to look to let science look even more amazing. So we take some venom cocktail, then we want to investigate something, and therefore we use a lot of chemistry, chemicals and physics, generate some ions, shoot them into a space with a high vacuum, then manipulate their flight with strong magnetic fields, and then use the power of the global databases and computer power to finally what was in our drink. So I think it shows how awesome science is, but since we can never stop, it's time to improve even this fancy method. And so I want to show you 3 topics we investigated, me and my collaborators, and what we can even improve, or what other questions we can direct to the snake venoms. So why all you can see here is measured, so in the background is measured to analyze the relative composition. So there's no quantitative amount that you can say so and so much molecule of that family was in, because it's just set to 100 and so give you the relative values. You can just easy quantify the toxins also in a simple way. But we can also simplify that graphic a bit. And let's say we start with the venom. We give some chemical ingredients into the measurement where we know the concentration. Then we use the mass spectrometry. And since we know how much we gave of this here in, we can then quantify our toxins that have some also the sulfur in, but that I will talk about in the next slide. And in the end, since we can now compare the known concentration with the unknown concentration that was in the same sample, we can absolutely quantify. And since it's Star Wars Day, I can either call it ICP, triple QMS, but you can also say it's more or less a lightsaber. So we have here a plasma torch that is ionizing your sample. And if you look a bit deeper into that lightsaber, then we start with a liquid. So with a snake venom that was separated over an HPLC. Then it goes to the gas phase, even to the atoms. We have here up to 8,000, 10,000 Kelvin, generate some ions that are oxidized. And in the end, everything is burned down to the atoms. And so since we added this compound or some similar compound, this had sulfur in, and we know that a lot of toxins has a higher content in cysteines or methionines, which also contains the only proteinogenic amino acid, the sulfurin. And with a mass spectrometry, we can here directly in the end quantify how much sulfur we had in our sample and compared with the known concentration, and then that way calculate the, we can directly quantify the venom for that toxin family. And the snake toxins need that or need most of the sulfurs to make sulfur bridges. Like in small peptides, there are often 1 or 2, but also bigger ones like the phospholipases can have up to 7 or 8 disulfide bridges. So the only drawback is you have to know how many cysteines your toxin has, and where it also elutes from the HPLC, but therefore we can implement a split, and then we have two graphs where we directly can compare the retention time from our lightsaber with the retention time for the identification of the toxins. And that's one way to use it. But what would be Star Wars without even ASTA? So we can also analyze our snake venoms by laser shots, and therefore we use MALDI. So we have really a laser that is pointing with a aiming cross on a specific point on our plate. We use a special matrix where our fraction of the snake venom is co-crystallized, and then we get a nice spectra. We can directly annotate the sequence of the protein that were, peptide that were in our sample or in the peak that we spotted on the plate, like here for some Kunitz inhibitors, phospholipase. But we can also go up to bigger proteins like CRISPs, the reproteases, and even super big proteins like the metalloproteinase of 50 kilodaltons or even 70 kilodaltons could be identified with this method. And what is better than one laser? Of course, we can just use to investigate something like the big plane. So here we have a cut of the venom gland, and better than one laser is, of course, a lot of lasers. So we shoot more often on our sample, and this is called MALDI imaging or mass spectrometry imaging. And so we aim with our laser on each spot here on the gland and rasterize so our sample and get so for each of the different points a specific spectrum, and that we can then analyze to say, here is that and that fragment, here is that and that fragment, and so on. And if we now look into the results, then you can nicely see that a lot of different toxin families are also stored or produced even in different parts of the gland. Here for Cobra, and you see that the metalloproteinases has still some nice area where they are highly produced, while, for example, the phosphodiesterase seems to be just mostly abundant in one part of the gland. And so you see that even within one gland, the variation of the different toxins can be seen, and where all these toxins are stored within this gland. And on the next level, even different proteoforms we were able to investigate here on the three-finger toxins. And here you see that different areas of the gland also produce different types of smaller peptides. And with this, I think, or I hope I could show you some about the amazingness of vipers and how we can investigate their venoms. And a special thanks to Benjamin, who I think is also in the audience, who was my supervisor even in the master thesis and guided me in this project, and to all the other collaborators, and especially the snake venom collectors, because without the venom, I could not make anything with our fancy machines here. Yeah, so thank you. Therefore, I'm done.
Clapping for you, Mark. It was excellent. Thank you for being so on theme with Star Wars Day, because I was thinking, shall I let you email you and Ivan to say we all need to dress up? But this was great. This is a useful acknowledgement. Before people ask questions, I'm just curious. Before you started your PhD, were you already into venoms, or did you start working on venoms at the beginning of your PhD?
I even started the venom fascination at my A level. So at the last year of my A level, I made there my final presentation of the hemo and cytotoxins of snake venoms. Wow. Or even on viper venoms, it was there. And then I started biochemistry, and then I could regain the focus to combine my interest in the snake venoms together with the biochemistry, and then make my master thesis about the viper composition and state. Now with it, and I think I will stay forever with the topic.
Well, that would explain, because you're quite prolific already at this stage in your career, so that's an indication of this. Dear audience members, if anybody has a question, this is the chance for you to engage Mike for a few minutes. You can either type your thing, or I can see that Siddharth Bhatia raised their hand. Siddharth, if you have a question, you can speak up.
Hi, thank you. Mike, I just have a question on your last slide. It was really fascinated because so you've shown these venom gland, and you've shown that all these venom toxins are being segregated. But in the venom gland, this is basically a liquid sort of mixture, right? So all these toxins can diffuse to different parts, but it's not happening. Why is that?
We use the gland of a cobra where you don't have really a big lumen where the venom is stored. So in the glands of the vipers, you have in the middle one big lumen where it's stored, but in the case of the cobra, there it's more, or in the elapids, it's more stored into directly the cells. So, and if you zoom, or if you would zoom here in the stained in the stained sample, and then you see that the cells are even really huge. So I think in the row from here to here, you have just 4 or 5 kind of cells. So there's an arrangement of a lot of secretory cells that formed one single compartment, and then it's stored within there. And so we were able to identify this kind of liquid while stored in this arrangement of cells, because there's no central lumen like in the vipers where it could be directly be washed out.
So it's like different cells are producing different toxins.
It's, at least in our sample, it looked that like, yeah. So we have some areas with different kind of cells that produce different kinds of venoms.
And did you do a temporal study of this? Like, did the cells change what they were producing?
And that we don't know. So until now, we just investigated this one gland, and we will see what the future of investigation will bring about that.
Yeah, that's very interesting. Yeah, thank you.
Thank you for your question, Siddharth. There's a question in the a two-part question in the chat. I will read the 2 questions out sequentially. Great talk. This is by Ferenc Turek. Great talk. I have 2 questions. Xanthina can be found on Greek islands. Do you know about any intraspecific variations?
No, I don't know, but I would like to.
Okay.
So I think as soon as you mentioned interspecies differentiations, I'm always in. But until now, I just have this one venom from a Turkish population. But of course, it would be awesome to have a lot of different ones for all of them.
Yes. Second question that Ferenc asks is, for selenoglyph snakes, do you focus on the accessory glands?
And not until now. Until now, we have just investigated here the main gland of the cobra, but in future project, we also will investigate the accessory gland.
Cool. In the interest of time, and also to let you off the hook, Mike, thanks very much. That was a beautifully presented talk with beautiful illustrations. Siddharth, you can lower your hands because unless you have a new question. So thanks very much. I'm going to stop recording now.